Hello, everyone. I hope you can hear me. I'm talking to you from my shed in South London, in the back of my garden, and excited to present to you all. I've got my slides up on the share screen, so you should see that. But if you look at the video as well, you can see the slide behind me as well. I might point using my hand or with my cursor. Hopefully this will work okay. So my research group is in the Bioengineering Department at Imperial College London. You can go to our website and read about our research, which is quite diverse in lots of different areas, but all in the theme of synthetic biology, where we recode with DNA. And so you can see in a bit more sociable times than what we're in right now, my group and some ex-members of my group getting together for Christmas earlier this year. So what we do is we research methods and try to generate new approaches in synthetic biology. So synthetic biology is obviously one of the breakthrough subjects of this century so far. It's really about applying engineering principles like modularity, measurement, and computer-aided design to biology. And it really can be summed up in the most simple way as treating DNA as reprogrammable molecular code. So thinking of the DNA inside cells, and to some extent as well the RNA, as the code, like code inside your computer that runs programs, and thinking about how to learn how to write that code and reprogram the code so that we can get the cells to do new things. Now, biology gives us a helping hand because the code is ATGC, so it's pretty similar to binary, zeros and ones, but also that biology naturally uses modularity in its code and programs for directing the cell to do things. So every gene typically has a control element upstream of it, which is the promoter, then the functional element, which is the coding sequence that creates the protein, which typically becomes something like an enzyme or a structural component. And then you would have insulation elements as well. So similar to how lines of codes in a computer will have the header that describes when the code will run, and then the main body of the code, and then the footer that ends that code running. We see that naturally in genomes. And really synthetic biology is just about taking this principle and doing it in a kind of a man-made way that allows us to write new programs. My research group really tries to push modular engineering in lots of different ways. Our four main research areas is understanding how to most efficiently engineer cell resources to go towards biotechnology products or for cells to do new tasks. Using modularity of genes and promoters and other DNA parts for tuning pathways, both metabolic and signaling pathways. Growing functional materials by programming cells to make materials in response to certain conditions and functionalize them as materials. And we're also involved in projects that are building synthetic genomes from synthesized DNA, not just small programs of DNA, but now whole chromosomes for cells. We focus, because it's the quickest and most tractable way to do things, on microbes. And the predominant work in my group has been over the last 10 years in Baker's yeast as cerevisiae, which is also the focus of this talk, but also in E. coli bacteria. But we also have projects that work with B. subtilis bacteria and Geobacillus bacteria, as well as K. raticus bacteria, which is a bacteria isolated from kombucha that produces materials, and Pichia pastoris yeast, which is an alternative to Baker's yeast that's a bit more proficient in protein overproduction. But Baker's yeast is our favorite of these cells. Apologies to those in my group who work on the other ones. But yeast really is a kind of a magic organism to work with. And it's been a magic organism for many centuries. It was for years thought of as a magic ingredient, something that the heavens provided for us, that somehow magically made a brew ferment and produce alcohol. And it's this use of yeast in the production of alcohol and in leavened bread over centuries, if not millennia, for humans, that really means it's an organism we know how to work with and understand how to work with. It's obviously got a strong history in food biotechnology with wine, beer, and bread production, as well as many other things produced from it. And right now, in this pandemic, particularly during the lockdown, a lot of people have been starting to get their own experience with yeast by doing things like trying to grow their own sourdough starters. You can see a disastrous sourdough starter in the slides here. This has got to sort of mean proportions in terms of how many people are growing their own sourdoughs, that XKCD have came up with this lovely proposal here that perhaps the coronavirus is simply a yeast symbiont with an extremely convoluted parasitic lifestyle that has allowed sourdough starters to spread all over the world, thanks to the way the coronavirus has locked us all down. Yeast is also something that's used in biotechnology, and particularly in medicine. Since the mid-1980s, the predominant supply of the world's insulin comes from Baker's yeast. Recombinant production of human insulin from yeast took over from the initial E. coli production in about the mid-1980s from work done in Denmark by Novo Nordisk. And lots of other drugs and biologics are made by yeast. Yeast is also a real focus in biology research because it's a very simple eukaryotic organism that can do a full sexual life cycle and can evolve and be traced with very minimal amounts of equipment, very minimal growth media conditions, and in a very short period of time. So it really provides a fantastic organism to use for the study of biology. You can see some fancy equipment here on this slide. Automated mating in very high throughput numbers can be done with Singer rotor machines, and fermentation with yeast can be done to brew up metabolites and to do that in very controlled conditions. Yeast really works out as a really good, awesome organism for synthetic biology, because as synthetic biology was arising about 15 years ago and starting to think about how we could engineer cells, it was able to already link in with an established pipeline of work in yeast biotechnology, where people were looking to turn sugars, particularly waste sugars, into products, primary, secondary metabolites, recombinant proteins, fuels, using biotechnology approaches. And this partnered really nicely with the fact that Baker's yeast was also one of the top model organisms being studied in systems biology, another reasonably new subject that has been a phenomenon of this century since we've started to get genomes and transcriptomes for many organisms. So yeast is really well positioned for synthetic biology work. And what really makes it a dream to work with compared to many other organisms is the fact that the community who do yeast synthetic biology have produced some brilliant tools that allow us to plug DNA code together, like we're putting together pieces of code online, or putting together pieces of DNA, pieces of Lego together when we're building new ideas. So we in my group use a modular DNA toolkit developed at UC Berkeley, which uses Golden Gate-based cloning to enable us to very quickly, in the space of a week, build up some quite complicated DNA programs where multiple genes can be expressed and can interact with one another. And it's really kind of, in some respects a bit like child's play. We're picking and choosing DNA parts from a catalog of things like promoters of different strengths, promoters with different regulation, and mixing and matching them with different downstream genes, whether those are genes that make the cells fluoresce or change their regulation. So we can typically build hundreds of plasmids very quickly with this system. Okay. Someone's put something in the chat. Good. Okay, so we can go beyond that, not just building these modular programs with just a few genes, but us and others around the world are working on using synthetic DNA and building this up in modules to make an entire synthetic version of the yeast genome. This is called the SC 2.0 project, the aim being to replace all 16 chromosomes plus the mitochondrial chromosome of Baker's yeast with versions that have been made from synthetic DNA with a variety of design changes. It's a big community project with teams around the world. Already 7 chromosomes have been completed and published, and the other 9 remaining chromosomes are all very close to completion, if not completed, but just going through the final stages of bug fixing and publication. My group joined this several years ago, taking on chromosome 11 for this project. And we completed that a few years ago, building up this 670,000 base pair chromosome of synthetic DNA inside the yeast, whilst deleting the wild type version of this chromosome. And it can replace all the functions of the native chromosome, despite being made of synthetic DNA with a lot of changes. A lot of the changes we made did lead to bugs. And so over the years, Ben Blunt in my team, who's since moved on to Nottingham University to start his own group, did a lot of bug fixing, where some of the design changes we'd made to the yeast chromosome 11, had led to fitness defects. But almost all of those are fixed now, so that we have a synthesized and fully working version of chromosome 11, which we can then add to the other set of synthesized chromosomes, hopefully to be able to complete the synthetic yeast genome within the next year or so. There's lots of different features of making the synthetic chromosome compared to the wild type chromosome, with things like introns being removed from the genes when they're not essential, having all transposon sequences removed from the synthesized genome. But I'm not going to really go into the synthetic yeast project in too much detail in this talk, because that's not the focus of this talk. But I just want to mention that probably the coolest trick that we're adding to the synthetic yeast genome is a system where the chromosome can shuffle up its genes on demand. So this slide briefly tries to show you this. As we've synthesized the chromosome, immediately downstream of every non-essential gene, we have put a LOX recombination site, a version of it called LOXP-SYM, and so that's highlighted with these green dots. And so the cell normally just grows and functions fine with the synthetic chromosome version, and these recombination sites just sit there and nothing's happening. But if we then add a plasmid into the yeast cells that expresses in an inducible fashion the recombinase Cre, Cre will then shoot into the nucleus when the inducer is present, and it will bind to two or more of these LOX sites, these green dots in the chromosome, and catalyze recombination reactions between them. And these will lead very quickly inside the cell, within minutes, if not hours, to rearrangements such as translocations and inversions, and deletions of regions of the chromosomes, and even duplications and insertions as well. And so this means that quickly after you've induced this system inside a cell which has a synthetic yeast chromosome, probably within an hour or two, many of the yeast within the pool will have diversified their genetic content by shuffling around the genes in the synthetic chromosome, deleting some out and duplicating them. So it's a really, really fast-paced evolutionary system that we're able to induce. And we call this system Scramble. It's kind of like shuffling the cards to get a different set of cards if you're trying to win at a game. Okay, so that's just an overview of the kind of technologies we're using in synthetic biology in yeast in my group. But a few years ago, we obviously began some projects that were a bit more applied, trying to solve some real-world problems. And the one we thought of and attempted first, probably about 6-7 years ago, when we sought the funding for this, was trying to use yeast synthetic biology to help with the antibiotic crisis. And so, in that regard, we decided to see, can we make yeast cells start running metabolic programs that produce antibiotics, and then maybe we can evolve them to produce different antibiotics, and in theory, maybe be able to produce some new antibiotics. So the first thing we wanted to try and achieve was antibiotic production from yeast. And so we decided to be inspired by work originally at the sites of Imperial College, which discovered the famous antibiotic penicillin. Alexander Fleming's work at what is now St. Mary's Hospital found this classic beta-lactam antibiotic, which is very widely used and has saved millions of lives over the years. This is produced by Penicillium chrysotium, which is itself a fungi, as Baker's yeast is. So we thought that this was a realistic thing to try and achieve. The pathway for making benzyl-penicillin requires a few steps here. And Ali Awan, a postdoc who joined my group but has since left to work on nanopore sequencing for the NHS, tackled this problem over a 3-year period. And I'm going to summarize now over the next 10-15 minutes his work and achievements in this. So the pathway requires bringing together 2 normal amino acids and a non-standard amino acid to make first a beta-lactam backbone structure, isopenicillin N. And then this is then modified through some metabolic steps to produce penicillin G inside the P. chrysogium cells. You can see the pathway here with the latter stages of the pathway occurring in the peroxisome compartment of this eukaryotic fungal cell, and then the penicillin G being exported out of the cells to go off and kill the local surrounding bacteria. The system requires 5 genes to be able to work, and you get the longest one, a 12 kb gene, is the real important one at the beginning. And that is a type of gene, a type of enzyme called a non-ribosomal peptide synthetase. This is a class of enzymes which are considered what they would call like assembly line enzymes, which are really kind of like a fusion of lots of different enzymes into one big enzyme, which catalyzes a lot of steps. And it catalyzes the bringing together of the valine, the cysteine, and the non-standard amino acid, the third one, to create the backbone structure. And a second enzyme, NPGA, then does a final modification to release the basic beta-lactam backbone here. So we got these genes and coded them into the genome of our S. cerevisiae yeast and checked that they could make this beta-lactam initially. But then we needed to then build up the metabolic pathway to convert this initial beta-lactam into the penicillin G. And so that required cloning these 3 enzymes from P. chrysogium into a format that can be expressed in S. cerevisiae yeast. And the last 2 of these 3 needed to be tagged with a modular tag that would then send them to be expressed inside the peroxisomes of S. cerevisiae, similar to how those enzymes work and are expressed and then work in the peroxisome of P. chrysogium. When we first built the first 2 genes, we were able from mass spec to see that we were producing the beta-lactam backbone structure, small amounts of it in our cells compared to a standard. And our first initial design with the full pathway with the 3 further genes, including 2 in the peroxisome, we could then see small amounts of penicillin being present in our cells compared to a standard here, with LCMS being used as the standard measurement here. Now, if you remember earlier on, I was talking about the beauty of our modular cloning system allows us to pick and choose which parts we combine together. So we were able to put together a variety of different promoters with the different genes and put together different terminators, or also known as poly-A signals, with those, and build this up into a library of different options. And what we're trying to do here is optimize the right expression levels of each of these 3 enzymes to ensure that we've optimized the flux of the metabolite, the beta-lactam backbone structure, being converted into penicillin. That could be simply just having strong expression of all 3 enzymes, but actually if one of those enzymes is not a bottleneck and you express it very strongly, then you're just getting the cell to do a lot of work for no extra gain. So it might in fact turn out that only one of these enzymes needs to be expressed strongly and the others can be expressed at a weaker level, and you would get a more optimal production of the penicillin. Ali used our modular cloning system to be able to build all the combinations with 10 different strength promoters in one go using Golden Gate in just a few days. And so that DNA assembly method gave us 1,000 different combinations, which could be transformed into the yeast cells that are already making the backbone. And that allowed us then to see 1,000 different combinations of promoter strengths for these 3 genes and measure using LCMS to find out which of those combinations was best. And we would see increasing increasing production levels of this penicillin compound inside our cells, particularly as we move to having the very strongest promoters for the 2 final enzymes, the ones that need to go to the peroxisome, and a medium sort of strength promoter for the first enzyme, the one in the cytosol, that gave the optimal flow. Now, the yields weren't great, but Ali managed to understand that our yields inside the cell of the penicillin weren't that great, but actually outside the cell, they were a lot better. You can see this here on the bar chart to the right here, that the penicillin inside the cell pellet was not at a very high concentration, but now we were seeing a lot more in the supernatant. So this was really exciting because we weren't sure how we would get the penicillin to be secreted by our cells, which is the aim that we wanted. But it turned out that it was just naturally secreted from our cells through some mechanism we still don't fully understand. And so at least that simplifies things for us. It means our cells, when they're growing, doing these reactions, they are producing the penicillin and it is gathering in the spent growth media, which makes it easier for us to use it. We were keen then to see how much penicillin we were producing and really check that it works as penicillin. And so we were able at that point, from the LCMS data we thought we were in bioactive territory. So we had enough being secreted into our growth media that it should be that our growth media should have an antibacterial property. And sure enough, we set up an assay where we would take our cells, do the growth to make the spent growth media containing the penicillin, and then we would place that on S. pyogenes bacteria and leave them in that media overnight and see whether there was an inhibition of growth compared to controls with just normal growth media. And yes, sure enough, we see a reduction of growth when we have the spent growth media from our yeast, and that is the equivalent of about 3 nanograms per mil of penicillin done from a standard curve assay. So this is pretty cool. As far as we were aware, this was the first ever demonstration of Baker's yeast being engineered to produce a bioactive antibiotic, and to be able to just also secrete that so that it goes into the growth media and sterilizes it, killing off the bacteria in that media. I mentioned earlier that we have these synthetic chromosomes for yeast, and we can do Scramble to do evolution of those. And so following on from the study that Ali wrote up and published in Nature Communications, as part of another study, we had the penicillin pathway inside a yeast cell with a synthetic yeast chromosome. We flipped on this Scramble system and then searched for strains that had altered the chromosomes, shuffled around the genes that now produced more penicillin. And straight away we found one that had double penicillin yields compared to our previous one from this adaptation, this shuffling of the genes, in this case in synthetic chromosome number 5 in the yeast genome. And this was part of another publication in Nature Communications led by Ben from my team a couple of years ago. So we were pretty excited by being able to achieve the first bioactive secretion of an antibiotic from Baker's yeast, which is this workhorse in synthetic biology. This was picked up by a few news channels, and they kind of simplified the story a bit and twisted the aims. So here you can see on the left here, the Times saying that beer holds the key to new antibiotics, which I think would be great, but I really wouldn't want people drinking beer which has small amounts of antibiotics in it, because that's probably just a recipe to get antimicrobial resistant gut bacteria over time. But it did make us sort of stop and have a pause on this research and think about what it's really useful for. At the beginning, what we were keen to show is that yeast can make these beta-lactam based antibiotics. And obviously penicillin is not one we need in large yields anymore. It's very cheap to get, and it's been a workhorse antibiotic for many years. What we would be keen to do instead is to get new antibiotics from this system. Now that could potentially be done by putting in different enzymes into our yeast cells and from more exotic fungi, and seeing how they modify the beta-lactam backbone, and maybe they will make different beta-lactam antibiotics. But the basic beta-lactam antibiotics themselves are kind of a class that plenty of variants have been produced over the years, amoxicillin, obviously, as well as penicillin and all sorts of others. And these are also a type of antibiotic which there's a lot of resistance against already. And I was quite humbled by attending some of the early meetings of the Antimicrobial Research Collaboration at Imperial College, the ARC initiative, to hear some of the other work being done by other teams around Imperial College. And it really made me think of this kind of frontline statement of that there's a crisis and we need new antibiotics is only a very tiny part of the whole issue. And some of the work people are doing about changing behavior, changing use of antimicrobials, or looking in particular at things like antifungals and the rise of resistant fungi, made me think again about the research area we were working in and what else can be done. In particular, I was very interested in that ARC meetings early on about the problems of pathogenic yeast and the rise of resistance of pathogenic yeast in hospitals and in healthcare. And it made me think of some work we were doing in our research group in other areas that was about detecting things using yeast synthetic biology and whether that could be put to task in this area. And so for the next part of this talk, I'm going to cover some of our work on yeast as biosensors and show you how yeast biosensors can be made to detect pathogenic microbes, and particularly pathogenic yeast that are just as much a problem as pathogenic bacteria in hospital and healthcare settings. So over the years with our modular toolkit, we've not only been engineering yeast cells to produce things like metabolites or materials, but to sense things as well. And particularly one initially PhD student, but now a postdoc in my group, Will Shaw, has done a fantastic amount of work sponsored by BBSRC and in collaboration with AstraZeneca at engineering yeast so that they can be used as reprogrammable biosensors for things in the environment. And so this is work now over the next set of slides, which I'm going to summarize, work that was done by Will with co-supervision by Mark Wigglesworth at AstraZeneca and with some help from a pharmacologist, Graham Lads at Cambridge University. So Will came to me, to my group, even before his PhD, with the proposal of engineering yeast synthetic biology, using yeast synthetic biology to engineer yeast cells to sense through G protein coupled receptors. So I'm sure many of you have heard of G protein coupled receptors or GPCRs. These are the largest superfamily of cell surface receptors. They're the main mechanism by which most eukaryotic cells, particularly the cells inside our own bodies and other mammals, detect what's going on around them. We have about 800-900 GPCRs in the human genome, and they can detect things as tiny as a photon, because some of them are activated by light, and as big as entire other cells, because they are also involved in sensing of touch and the touching of different cells, as well as more famously being there for the sensing of things like metabolites and olfactory molecules. And we have hundreds of these GPCRs in our nose and our taste buds. They detect all of those interesting things. Now, that huge variety of what they can detect is quite a contrast to the very limited structure that they have. They all have this very similar module in the middle of 7 transmembrane helices. So it's amazing that with this same motif in all of these thousands of different GPCRs in nature, they can sense so many different things. So Will was very excited about trying to be able to use this natural modular system, which is so diverse in what it can achieve, in synthetic biology. Now, yeast, Baker's yeast, which we work on, has 2 GPCRs, only 2. It's one of the most simplest eukaryotes and obviously one of the most simplest in terms of things it senses. It has one that senses sugar, so senses food. That's very important in life. And then it has the other one that senses sex, right? So it senses the pheromone produced from the opposite sex of the Baker's yeast. And so this is a very well-studied GPCR signaling pathway called the yeast pheromone response pathway, where the 2 different mating types of Saccharomyces cerevisiae yeast send signals out to one another called pheromones. These are small peptides. And they have GPCRs in their outer membrane that respond to these peptides and signal through a cascade of phosphorylation reactions through a G protein and then some kinases, a MAP kinase cascade, before activating a transcription factor to turn on hundreds of different genes that are involved in the mating response. And you can see on the right here some of the earlier steps of the mating response, which is that the 2 different sexes of yeast cell will shmoo towards each other, so they sort of project towards one another and then fuse together to make a diploid before then undergoing meiosis and sporulating to make progeny. Now, this classic pathway has been studied many, many times before and has been known for several decades. And for at least 2 decades, if not longer, it's been realized that you can swap out the GPCR that recognizes the pheromone for another GPCR from a different organism that recognizes some other ligand of interest. And as long as you start to delete out some of the other genes or somehow disrupt or inhibit them, the ones involved in cell cycle arrest and triggering all of the hundreds of mating response genes, you can instead redirect this signaling pathway to a promoter that then can be used to drive a reporter gene like GFP. And this allows you to build a sort of a simple biosensor here, where your GPCR detects its ligand of interest. And if you're lucky, all of this system will interface with each other and turn on expression of GFP. So now you have a system where your yeast cell, in response to the ligand, will produce GFP. More recently, people in synthetic biology have been making use of this system as a biosensor for things that we want to make. And Pamela Peralta-Yaha's group in the US, for example, put in an olfactory receptor that can smell or sense decanol, which is a medium chain fatty acid, which can be used as a biofuel. And they had that signal through the cascade to a transcription factor that produces GFP. And now they have a yeast cell they can use to sense whether they're making this biofuel molecule from their other yeast. And so that can simplify their workflow. So Will was inspired by this kind of work and wanted to be able to take this to the next level so that it could become quite a rational way to program a yeast cell to sense lots of different things. So the first thing he set about doing was taking the yeast cell and the wild type signaling pathway through the GPCRs, which you see on the left here with all of its complex parts, and stripping it down to make something much more easy for us to understand by deleting out all the things that interact with it that complicate how it behaves. So you have some factors that they interact with it, and if they get activated, they will feed back or they will change the cell's growth. That's not great if you want a rational, tunable sensor. You want it so that just the amount of input you get equals the amount of output, and you don't want complications from other side pathways. So Will set about making a yeast cell with a minimal signaling pathway that could be easily reprogrammed. These are all the things he looked at that are part of this pathway, all the genes, and he looked at which ones needed to be kept, which ones could all be deleted, and you can see many being deleted here, and then which were the key ones that we would want to be able to kind of reprogram and change to be able to get this sensing pathway to sense the kind of molecules we want and also be able to tune how well they sense them. So you see three key genes of the pathway need to be what we would say refactored, which means they need to be built from the bottom up using synthetic biology parts so that we can recode them to behave like we want. So to set up to get this minimal cell that can do this sensing required deleting a lot of genes, and Will built a fantastic little toolkit using CRISPR-Cas9 deletion cycles, deleting 3 genes at a time, each transformation, and then cycling through that through 3 selectable markers at a time in rounds of deletion that led to 9 deletions in a week, and used this system to remove 13 genes from around the yeast genome that we didn't want to be interfering with our pathway. All these genes are cleanly deleted from the start codon to the end of the stop codon, leaving behind just a landing pad, a barcoded landing pad that we can use later in the project. So you can see here from nanopore sequencing, we could verify that in our strain, we managed to do all the deletions in the different positions around the 16 chromosomes as desired. And our strain was now, even despite all of these genes being deleted, grew just fine in normal glucose growth conditions. In fact, it actually significantly grew faster than normal cerevisiae yeast in these conditions. So it was slightly fitter in exponential growth. We believe that's because by deleting out the components that are normally required for being poised for mating, we no longer are wasting any biomass of the cell or energy of the cell in thinking about mating in the future. It's not a requirement in this condition, so the cells can grow a bit faster. all of these deletions meant that we could have a base strain for modular refactoring of GPCR sensing. So simply put, we have this strain with all of these deletions, and we have the key genes that we need to put back in. The GPCR, so that's the receptor that's going to do the sensing. The G protein, the first protein in the signaling cascade that couples with the GPCR. And then downstream of this signal cascade, we need a synthetic transcription factor that then goes to a fourth gene, which is has the promoter for this synthetic transcription factor, and then a downstream reporter. And in most of our cases, we report with GFP, and we just track the green fluorescence of the cells using flow cytometry or a plate reader to be able to see how much output we have through this system. So in a simple, straightforward experiment, Will can build using our modular toolkit and Golden Gate all of his desired combinations of these 4 genes, these three genes plus this gene and its different promoters, into a plasmid, stick it into this cell, and he will have built a GPCR sensing pathway to his preferred design. So the first time he did that, for example, we build in a promoter in each of these positions for these different components, put them together, put it in, and he tried to recreate the system, the natural system, which responds to the pheromone peptide. And sure enough, this would be the dose response curve for increasing amounts of the pheromone versus how much output you get from the system, the signaling, the GFP output on the y-axis. And the gray would show you the normal response. So you'd have this nice dose response curve at around nanomolar concentrations of the pheromone. A first naive design with just constitutive promoters of different strength gave us this dose response curve, but a really poor dose response curve if you look at it, right? So it doesn't activate very high, and it now only activates at a 10 to 50 times higher concentration of the pheromone. So it's not ideal. And this is obviously going to happen first time you do this, because it's going to be hard to beat what evolution evolved over millions of years just in a first design. But Will realized that with his modular system, he would be able to tune this dose response curve through rational approaches. And this is really important to be able to have control over a dose response curve, because whenever you're making a sensor, you really need to be sensing in the right kind of concentrations for what you're interested in sensing. So there's no point sensing something in the micromolar concentrations if it's present in only ever found in millimolar concentrations. And also what you want is a good dynamic range and low leakiness, so that your system goes from, let's say if GFP is your output, having no GFP to having a large amount of GFP when you have the right, the signal present. And so we really want to have control over the tuning of the operational range, the tuning of the dynamic range and the leak. And ideally as well, it'd be nice to have control over the hill slope, so you can either have a very quick increase in output as the concentration of your input goes, or you can have a very slow increase so that you can then correlate how much, let's say, GFP expression you have and be able to work out that that equals a certain amount of input. So you can see the desire is to really have the bottom two here on the right kind of curves with very low leakiness and high output, and either a gradual increase or a switch-like increase. And all of these other dose response curves are really not desirable, because they don't really tell you whether you've got the right amount of input and have a strong signal. Now, we turned here to a collaboration with modeling team in Cambridge to be able to solve how we could tune these dose response curves to get into really good bounds, good dynamic range, good operational range, so that we can sense the right things at the right concentrations. Now, the modeling, if you've ever done mathematical modeling of gene networks, is typically done by saying all of these components are at a fixed concentration. Let's see what happens if we take one of the components, like one of the proteins in this complex, and change its expression level. So they would just give it a different number for the amount of it in the model, and it go up, and then they will see how that behaves. Now, because in this project, Will has effectively stripped out all of the things that interact with this pathway, and all of the key genes in this pathway are now expressed simply from modular constitutive promoters, we can do the kind of thing that is done in the modeling, but now in the cell. We can take one of these protein components and say, let's now see how the system behaves if that component changes its expression levels over a variety of levels. And it really works out nicely that the modeling and the experimental data can match up nicely. And this allowed us to come up with some model-based rules that then played out perfectly in the lab inside our cells. So rule 1 was that if you increase the concentration of the receptor, it increases the sensitivity. And so the model shows the dose-response curve goes up and to the left with increasing receptor concentrations. And we see that happen exactly in the experiments as we increase the promoter strength of the GPCR in our system. Rule 2 was that you wanted to increase the strength of the G protein, the first member of the signaling pathway, up to a level, but not too far to overwhelm the system, to give you a sweet spot where you can reduce the leakiness out from your dose response curve. The models showed it was at about 2/3 strength, and that matched up quite nicely with the experimental data we got. The third rule we got was that to increase the output, how big the range of the output signal from our system was, you wanted to change the synthetic transcription factor and the promoter in front of the GFP. And so Will built a library of synthetic promoters and with transcription factors that would increase in response to the amount of signal coming through the cascade, increase the amount of GFP produced to really get up to very high strength signals if desired, even with low leakiness as well. So with these 3 rules, we could then refactor these pathways and optimize them like you would optimize something on a mathematical model. So we can see our initial pore design. We've then optimized by changing the G alpha protein expression level to remove the leakiness. And you can see the leakiness disappears. Then we tune the receptor level to bring the curve up and to the left to get it to be as sensitive as the wild type system. And then finally, we changed the downstream transcription factor and the reporter promoter to really increase the output range to get a very high level of GFP expression in response to the input signal. This gives us a lovely curve, at least as good as the natural curve, maybe even better. And so with these principles, plus the modular toolkit, it becomes very quick and easy to make new biosensors in yeast. So the optimization and learning all that took 3 weeks, but because of that, now in the space of a week, you can take a GPCR from another organism, put it into our our yeast that have been engineered, our backbone yeast, and straight away achieve really good dose response curves that are as good as those found naturally in the other organisms. And here we can see it from S. pombe yeast taking its sensor for its own mating factor and moving it over into Baker's yeast. So AstraZeneca were very interested in detecting metabolites relevant for human health. So the GPCRs we were bringing over and testing as part of this project and able to get lovely dose response curves for were ones that detect metabolites found in human blood. So we put in the human adenosine receptor, we put in a human melatonin receptor, and those work really well at making yeast cells that could sense these molecules. And with melatonin, we even went as far as showing that the yeast cells sensing the melatonin could do so with such strong specificity, not sensing any of the other precursors that produce melatonin in the normal melatonin production pathway, but also sense very accurately over a large range of concentrations so that they were as good as mass spectrometry detection. And the way we demonstrated this was using these yeast engineered to sense melatonin, to sense how much melatonin was being made by a library of yeast produced by a team in Denmark that are designed to produce and secrete melatonin. And so here we were able to put our yeast with their yeast and detect which of their yeast are producing the most melatonin. And the results correlate really beautifully with the gold standard mass spectrometry method of detecting the melatonin levels. So like I said, with AstraZeneca, and this was mostly focused on sensing metabolites using this toolkit. And so you can see some of the metabolites here we've been sensing with this toolkit. melatonin, adenosine, serotonin, glucose. And since then, and that work was published in Cell about this time last year, and our toolkit is shared widely, including our strain. A lot of people have requested it from AdGene for various sensing projects, both commercial and academic. And Will is currently reformatting this for sensing cannabinoid molecules as well, using the human cannabinoid receptors. But for the topic of this talk, what was interesting was that yeast that sense through GPCRs can also detect fungal pathogens. And we knew this because a group we've collaborated with in the past, Virginia Cornish's group in New York, in work led by Nili Ostrov and published in 2017, showed that Baker's yeast cells can be developed as a dipstick biosensor for a variety of different fungal pathogens. What they did was, using the kind of the crude mechanism being shown earlier on for biofuels detection, they programmed in GPCRs found naturally in different yeasts that recognize the mating factors of those yeasts into Baker's yeast, and got those to activate enzymes involved in a metabolic pathway that produces lycopene, the red pigment found in tomatoes. They then showed that these yeast cells could be put on kind of like little paper sticks, soaked in growth media containing suspected samples that might contain the fungal pathogens. And if those fungal pathogens are producing their mating factors and the yeast cells have the receptors for those mating factors in place, that should activate the expression of the enzymes in the pathway, and you should start to produce red pigment from this dipstick. A really nice idea. This was work sponsored by DARPA, and I believe they have a startup company working on this. But it's slightly problematic in the fact that the sensitivities are quite low, and also it takes many hours for the yeast cells to activate to produce enough of the lycopene to give you the red signal. And perhaps the most complicated part of all this as well is if you have some activation but not enough activation, what happens is your piece of yeast on a paper sort of starts to turn a bit red but doesn't get that red. So it's really not a yes/no answer. So it's not really that nice to use as a point of care diagnostic, because you sort of end up with a lot of gray area, or I should kind of say pink area, because it's between white and sort of yellowy white and red cells. So we used our toolkit then. We realized from the work we'd done in our toolkit, we'd done something very similar to what they did in that paper when we brought in the GPCR receptor from S. pombe yeast, put that in the Baker's yeast, and very quickly got a perfect growth, perfect induction curve where the yeast cells are now responding to the pombe mating factor. So in this demonstration, we'd already built a cerevisiae that very accurately detects where the pombe yeast is present. But the signal dose response curve was not was quite a slope. Let me just go back here. You can see it starts at -8 and goes to -6. It wasn't really the switch-like behavior we would like for a sort of yes/no point of care system. So we'll address that by modifying our yeast cells into a 2-cell system, where the first cell is designed to sense the thing of interest. In this demonstration here, it's sensing melatonin from our research paper. And then that cell secretes out yeast's own mating factor for the other cell to detect. And that other cell is producing a protease which deletes the alpha factor to maintain the sensitivity and setting a threshold. What you have here is a 2-cell system where one cell amplifies the signal, the other cell reports it. And this really narrows the range of concentrations within which you switch the response to the input. So you can see here the melatonin response going from a 200-fold decrease in the range so that it becomes a much more switch-like behavior. So with this system built, we could take some of the fungal pathogen sensing receptors, and I'll show you just the one example here. We took the P. brasiliensis fungal pathogen, the receptor for that, put it into Will's engineered yeast cells, And Ostrov et al. Paper showed this dose response curve, turning on more and more lycopene expression in the cells, going up to kind of micromolar scales of the peptide. But with Will's engineered cells and specifically the amplifier reporter two-cell system, we instead got a much more desirable dose response curve here, sort of a much more digital system, and with picomolar sensitivity, giving a yes/no answer here, going from about 10 to the minus 11 to 10 to the minus 10 with full signal output. So that's really a useful thing, and the Virginia Cornish's group has been looking at our approach for their company in the future. So to summarize, yeast is the foremost eukaryotic cell for synthetic biology, and we're using DNA engineering in this for a lot of different things, including making synthetic chromosomes. And over the last 5 or 6 years, we've been looking at using this kit for metabolic engineering in yeast, and we were able, as one of the demonstrations of this technology, to produce the first yeast strain that can secrete active levels of antibiotics into its growth media, and this was a beta-lactam. And we've been talking to people like the Kew Gardens Fungarium over the last few years about access to resources for other fungal enzymes that could be brought into this system to diversify these antibiotics. And then in the second part of the talk, I showed you that we've reprogrammed the yeast pheromone sensing pathway to be able to be used as a kind of a plug and play method to sense different ligands via GPCRs and have yeast cells report on that. And this is showing promise as a framework, these cells for sensitive point of use sensors for fungal pathogens. And we hope to be able to extend that further in the future. So I'll end by thanking sponsors, particularly BBSRC, AstraZeneca, who sponsored a lot of this work, but also some sponsorship from EPSRC and GSK as well. And particularly want to highlight the people in the yeast team within my lab who did a lot of the work described here, especially Will Shaw, who you see here, crouched down, Ali Awan, who did the penicillin work but left the group a couple of years ago, and Ben Blunt here, who's been doing the synthetic chromosome work with a bit of help from Glenn Gowers, shown here as well. Okay, thank you. I hope there's some time for some questions now at the end. I have the questions written up here, so maybe I can start to just read them out and answer them. Okay. The first one is there any cell encapsulation drug delivery work within SynBio research? There probably is. I'm not sure. We're not working on anything like that. So I'm not sure whether that means the cell itself is considered the therapy and that's encapsulated and delivered, or whether the cell encapsulates the drug and that delivers it. I'll have to pass on that one because I'm not really sure on that. Sorry, Ibrahim. Oh, done. Okay, so what is deletion and insertion? Okay, so in the Scramble system, let me go pushing back. The recombinases, when they bind to 2 lock sites either side of a gene or a couple of genes, and they catalyze the reaction, one thing that they might do is loop that DNA up into a little loop, which you see here. And then as the cell grows and divides, that loop's going to get lost because it's no longer part of the genome. So it gets that piece of DNA disappears. So we call that a deletion. The insertion would be if that or another piece of DNA is provided, the recombination could end up pushing that piece of DNA back into the chromosome, inserting it somewhere else through this recombination mechanism. There's 2 questions, beta-lactam, okay, let me answer the second one. A beta-lactam is a broad class of antimicrobials. You can see the structure here of penicillin, that's a beta-lactam. And so we call that because it has this kind of beta-lactam structure in the middle here, this weird structure. And basically penicillin is one example of what we would call a beta-lactam antibiotic. Amoxicillin is another one. All the ones ending in -cillin tend to be beta-lactams. So it's just something to do with the name of the structure in the middle of this molecule, and it's something found common in a lot of antibiotics. Okay, so Julia B asks, how do we select for the yeast cells that have effectively deleted the 13 desired genes via CRISPR-Cas and not deleted genes we wanted to keep? So this system set up by Will for the multiplex, the multiple gene deletion. The guide RNAs for the CRISPR only target very specific sites, and in yeast, because the genome is quite small, the specificity is very, very high, so it's very unlikely that you will have off-target effects. And you cut at these regions, and only the cells with the right selectable marker will survive. And so we check the selectable marker expression, so growth in the absence of leucine, then growth in the absence of histidine, then growth in the absence of ura. And after 9 genes being deleted in a round of these, will typically do PCR to check that those genes have been deleted. To verify that we didn't delete anywhere else by accident, we did this whole genome sequencing by nanopore sequencing to check that there be no deletions or rearrangements anywhere else in the genome, and that verified that all of these 13 genes had been deleted exactly and precisely as designed. So Antonio asks, are there different types of lock sites to control recombination of the different chromosomes separately? Yes, there are different recombinases, but in the synthetic yeast genome design, we are all using the same type of lock site. It's called LOXP-SYM, a symmetrical one, throughout all of the normal chromosomes, the 16 standard chromosomes that are being made synthetic. But there is an additional extra small chromosome that we're adding to the nuclear genome called a neochromosome that is going to house, or that is designed to house, all of the tRNA genes on their own, separate from the rest of the genome. And on that neochromosome, which is in completion from Patrick Tsai's group at Manchester University, instead of having the lock site, it has a variant called ROX, which is recognized by a different recombinase called DRE instead of CRE. And that allows that chromosome, which contains only tRNAs, to recombine separately to the rest of the genome, which will recombine using CRE-LOX recombination. Okay, Mayuri says, do you think you could detect SARS-CoV-19 using the pheromone system? Okay, it's something we've thought of. It's not something that's naturally sensed by a GPCR, but GPCRs can be evolved to sense things. And obviously, yeast that you can program and they grow fast, they're a very good starting point for directed evolution experiments. It's possible, but probably would be a challenge to get, outer surface fragments from COVID-19 and evolve them to and evolve a GPCR to sort of detect this. It's slightly complicated, or quite largely complicated, by the fact that yeast cells have a cell wall around them, which means that quite large things like a virus might not easily pass through the wall and then touch the membrane where the GPCR is. Fragments like peptides can, so you could theoretically lyse a sample and then detect whether GPCR, using a GPCR, whether a virus is present, if that virus is broken up a bit and some of the components can then be sensed. But the system still takes, a bit of yeast growth and a few hours before you get a signal. So I'm not sure whether it would be quicker than the kind of DNA-based diagnostics we already have. So it's not an area where we've pursued. Okay, Hannah asks, are there any other eukaryotic cells which can be used / are simple to use to secrete antibiotics? Yeah, so other yeasts would probably work. I would say Hansenula polymorpha is one we've looked at. That is a kind of cousin of Saccharomyces cerevisiae yeast that has large peroxisomes, and so holds a bit more promise for maybe producing larger amounts of things like penicillin. I can imagine that other fungi used in biotechnology, like Aspergillus and Pichia, or even some candidas, could probably also work in this way. I think the limiting factor really is the ability to be able to engineer the cells with DNA that can make them express the enzymes to make the antibiotics, and also the kind of underlying metabolism within the cells, and whether that is a good sort of metabolism inside the cells for making the antibiotics. In many ways, the metabolism inside Saccharomyces cerevisiae yeast is not very well prepared for some of the more exotic antibiotics to be produced, and therefore working with some kind of slightly more exotic yeasts and fungi, things like Aspergillus, for example, filamentous fungi, is possibly a better way to go for more complex antibiotics. How does beer hold the how does beer hold the key to new antibiotics? Well, beer holds the key for lots of different things, because you can have a drink with someone and then you can think up new ideas. That's a simple answer. I'm not sure how it does hold the key to new antibiotics. I think in the Times article, the headline writer just wanted to have something that brought people's attention to it. And then later on in the article, it said the yeast that is used for beer has been used in this study to produce penicillin antibiotics, and has the capability now to be used to plug in enzymes from different organisms that could potentially change the penicillin to make new versions of a beta-lactam antibiotic. Okay, next question. Could we have deleted all 13 genes at once using the multiplexing of the guide RNAs? Yeah, that's interesting. We have been looking at that. Whether that would be more efficient or not than doing 3 at a time depends on the work. So I think in collaboration with Rodrigo Ledesma Amaro's group, also at Imperial, our co-supervised students and Will from my group have been looking at multiplexing to express multiple guide RNAs in the same cell at the same time. And I think there are up to 20 different guide RNAs being expressed and working inside yeast in one go. And so that theoretically could delete that many genes in one round. But I think it's not that optimal. So only a few cells get it to work first time. And so there's a trade-off between, we know we can delete 3 genes in one round with a very high efficiency, something in this kind of 90% of all cells, 99, 90, 95% will do it correctly. And so then you can progress very quickly, whereas once you start to get to much higher numbers, only maybe 5 cells out of 100 might have done all 20 deletions at the same time. So you'll have to go and hunt for those cells by PCR, so it's not ideal. How have you and your team contributed to COVID-19? Hopefully not by passing it on to other people. We've not been in the lab, and so we've broken some theoretical chain of infection. One person has remained in the lab, which is Will Shaw, the guy who's done all of this work. And he has basically seconded to stop working on all of his current projects and instead try to express the spike protein receptor binding domain from the outer surface of the virus, express that and secrete that from the yeast cells, so with a modular tag attached to it, so that it can be then covalently bound onto a particle, and this would then produce a viral-like particle that could be used as a vaccine. This is effectively, he's doing the backup of a backup of a backup for Imperial's vaccine project. So Imperial has a vaccine that's in quite advanced stages based on DNA, but another research group at Imperial has a viral-like particle vaccine in going well through phases of trials in animal models that is produced using protein biosynthesis in mammalian cells. And because mammalian cells are quite hard to scale up to massive production scale, they've asked if our group and other groups like Karen Palizzi's group can help with seeing if the vaccine can instead be made by yeast, which are much quicker and cheaper to be able to produce vaccines from at very large scale. So that's how we've been helping out, although it's largely been Will rather than me stuck here in my shed. Okay, final question on this list. When you used the Scramble method to improve antibiotic production, were you able to pinpoint the reason of the improvement? A good question. So it's tucked away in our Nature Communications paper describing this. This is the work where we did a scramble. And so the chromosome that was scrambled was synthetic chromosome 5. What happened was it deleted and changed the expression level of genes involved in the cell cycle, 2 of them, I seem to remember, although the details aren't perfect in my head right now. What that then led to happen was that the 2-micron plasmid, which is the kind of plasmid that most of our pathway was housed on, that now increased its copy number inside the cells to roughly double the amount of copies of that piece of DNA per cell, whilst the cell is still growing at a normal rate. And that then meant that we doubled the gene expression of all of those genes in the pathway, which then led to double the yields. I mean, we'd already seen in the previous work that having the strongest promoters possible for the final 2 genes in the pathway, the 2 enzymes in the peroxisome, was the key to having the very highest yields. So this scrambled strain now allowing us to have twice as much copy of the plasmid per cell allowed us to basically double those expression levels, and that then doubled the penicillin yields in a very nice way. Okay, that's all the questions in the list. I don't know if that's where it all ends now. Hopefully I haven't gone too far over time.
Hello.
Hi.